Thanks y'all for inviting us here. We're just thrilled to be here. Full disclosure, there was an Arctic air mass up in Minnesota that just pushed us south, right? Our research team, and we timed it so that we're here to provide this presentation perfectly. Also, I got my master's in and PhD at Texas Tech, so that could be a really good or a real bad thing here. I understand that, but get your guns up. So yeah, I'll talk a little bit about our center, the Minnesota Center for Prion Research Outreach. And over the last 3 years, we've developed some really exciting technologies that we think are going to provide new options in the fight against CWD. And so really the majority of the talk today, I'm going to highlight some of those technologies because we think you all might be interested in those. Okay, so the center itself, it was established in 2019, and it really came about by, I was interacting, and other faculty members at the University of Minnesota, we were interacting with legislators in the state, and they were asking all kinds of questions, like, why are the diagnostic options so limited? Why can't we detect these prions in the environment? And they're just rattling these questions off, and I was always like, I don't know, I wish I had the answers. And so after talking with them, it was like, you know what, the only way that we're gonna really get ahead of CWD and be able to start fighting this war effectively is if we had a really focused research effort that was aimed to develop next generation technologies, aimed to develop new tools that would free up new options to fight CWD. And so that was the origin of MinPro, and it was from the start, it was thought of as a multidisciplinary think tank environment. And we put together we got an initial investment from the state about $2 million, and we put together this really advanced prion research lab in about 5 months and just started cranking away. And we have faculty members all over the university now, and then other institutions as well. But our goals were to develop next-gen, cutting-edge technologies to fight prion and protein misfolding diseases. That includes Creutzfeldt-Jakob disease, Alzheimer's, and Parkinson's. And again, new tools and strategies for CWD because we need that to effectively fight the war. And with me here today is co-director Dr. Tiffany Wolf, associate director Mark Schwabenlander. And again, we have like 40 research affiliates across multiple universities, state agencies, travel nations. And part of the reason we're down here is because we've had this great interactions with folks down here in Texas, including at the Cesar Kleberg Institute. And we really think there's some synergistic ways that we can collaborate on this disease. Okay, so outline for the talk today. We're fighting this war against CWD prions. I'll talk about what a prion is, because we hear about prions a lot in the news, but do we really understand what they are and how they can become infectious? And I'm a optimist. Everyone that knows me is like, I'm an eternal optimist, and I think that we can develop next-gen technologies that will help us win the war, not just fight it, but win it. And so I'm gonna highlight some of those towards the end of the talk. How many of y'all have heard about what a prion is, or heard about a prion? How many feel comfortable you really know what a prion is? Whew, right? Like that's how it always is, right? We've been hearing about this, and the part of this is because prion biology and the science of prion diseases is pretty young, right? I didn't get that at Texas Tech. That's not a limitation of the education at Texas Tech, but it just wasn't there, right? So it turns out all mammals have prion proteins. And this diagram, this movie here, is just a ribbon diagram to help us better understand what prions look like, right? So these are what proteins look like. So we all have normal prions in our bodies, in our cells, doing routine physiological activities day to day. These proteins help regulate copper, zinc, iron metabolism. Very important functional roles in nervous tissues. They're enriched along axons and at presynaptic terminals. And one way that you can think about this, okay, this molecule is so important for understanding CWD. The one way that we've learned that we can have a better understanding of this is a slinky. So this model, it came from my wife, didn't come from my brain. She's a great educator. So if you think about this slinky, it's a three-dimensional object, right? It flows like this, and I could go to the stairs and it would go down the stairs. You have to think in 3 dimensions when thinking about prions. This is a normal functioning slinky. It's a normal functioning prion. This is what's in our bodies right now. Okay, so when you think about normal prions, think about this slinky and how it's shaped. It has this normal function. One way that, let's see if this animation comes together. It looks a little different on this screen here, but one way that we can talk about prion function is looking at the cell. On the left is a healthy neuron. On the right is this zoomed in on this cell body here, but right now, after you had that lunch, what your prions are doing right now is they are on the edges of cells throughout your body. They're binding copper and they're bringing copper in. Hopefully this process is going to make you all tired and sleepy for the talk today, but that's what's happening. That's what they're doing in your bodies right now. Well, how does this get into something that causes CWD? How does it get into something that's infectious? The way that you can think of a CWD prion is just a misfolded, messed up slinky. If I were to go and put this down the stairs, it would just roll down the stairs. It's completely lost its structure. It's no longer got this three-dimensional structure that has a function. It's lost its structure. It's all messed up, right? It's all twisted. So that's like this infectious prion model on the right. We know that the prions that cause prion diseases like CWD, but also Creutzfeldt-Jakob disease and BSE and Scrapie, they've changed their shape, and they have these things called beta sheets that stack together and form fibrils. That's the abnormal form, right? So normal slinky, normal form, messed up slinky, abnormal form. That's one way that you can view this molecule. The three-dimensional structure is really, really important. You can also have different shapes. So if this messed up slinky has different structure, different shapes, they can be different strains. So Scrapie may look like this, CWD may look like this, Creutzfeldt-Jakob disease like this. So the three-dimensional structure matters. So when an abnormal, right, if this is our CWD prion, let's say a deer ingests this, it comes in. If this comes into contact with this normal prion, it will cause it to change its shape. Just think of it like it's coming in, it's literally like touching this physically, interacting with it, and it just like gets real relaxed and starts to misfold. That's what's happening kind of at the cellular level when deer are exposed to CWD prions, comes in, and then you start this daisy chain throughout the animal. And it takes about a 2-year process for that daisy chain, that domino effect, to work its way up into the brain, throughout the different tissues of the body. And then over time, it just starts to destroy, right, nerves. We know that these are enriched in nerves. It'll start destroying the nerves, and that's why you see the neurological effects of late-stage CWD. It's actually destroying nerves. These prions are resistant to degradation. When it takes that shape, those beta sheets, we know like from Scrapie, for example, some of those molecules can remain infectious in the environment for up to 16 years. Right? We know that research out of Colorado shows with mule deer that for sure 3 to 5 years in the pen, you can put mule deer in a pen and they're exposed to these in the environment, they'll get chronic wasting disease. So this is shielded. You have to think like when it changes its shape, it's resistant to degradation. That's why when folks talk about prions and why it's so important to keep CWD control is part of its environmental contamination, right? Because these can remain in the environment for some time. There's also research on Scrapie that shows that these can be detected on dust in old Scrapie barns. So that's the scary part, right? But CWD prions, they can be spread by both direct, like animal-to-animal contact, and also indirect routes, that environment-to-animal route. Okay, so when I talk about this in front of our legislators and to folks like y'all, I say like this, and I know the politics of CWD, it's a bloodbath, right? This is the enemy. How do we fight this, right? Because we have to have new tools that will effectively allow us to identify this thing, to neutralize it, to stop the spread of it. This is the enemy. So that gets us into CWD diagnostics. We know that the current diagnostic tests have lots of limitations. ELISA and IHC, these are the traditional tests. This is published, or this is like from 2017. Every year there's shortages. I think right now there's a shortage in ELISA kits. There were major shortages of IHC testing supplies last fall. Hunters up in Minnesota can wait like 4 weeks before they get the results when they want to test for CWD. These approaches, ELISA and IHC, they are expensive. They require large labs. There's a lot of limitations for them. right? And you can also miss early stage infections with some of these just because these are older technologies. They're really useful, but they're a little bit older, and you might miss early stage infections because they might not be sensitive enough to pick up, prions in an animal that's only had the disease for about 3 months or something like that, right? Lots of limitations there. So we need, if ELISA and IHC is the first generation of CWD tests, we need to look to a second generation. How can we move beyond those and develop a new class of tests that we can leverage to find these molecules more effectively? There is a second generation test that's available, and that what we have in our MinPro lab, and other labs around the US have the same technology. They've actually had it since like 2007. It's developed for Creutzfeldt Jakob disease in humans. And many of you are probably hearing more and more about RT-QuIC because there's a lot of interest in that, and it's currently undergoing USDA validation for CWD testing. But I want to dig into actually how RT-QuIC works, because there's also a lot of misinformation out there. But we can use a Slinky model to help understand how this RT-QuIC test works, and then to give you a better understanding, like when you hear about it in the media and whatnot, you can actually part of you will know how this works. So essentially what we're doing with RT-QuIC is if you have a test tube, let's say I'm a test tube, you fill that test tube up with normal prions that we can grow up in the lab. Like we can grow up recombinant prion protein using E. coli in the lab. You put those into the tube, right? Let's say this is a sample of a positive lymph node. It's a lymph node that's positive for CWD, right? You just take a real small speck of that. You seed it, your reaction. You take this and you put it in that test tube, right? And it comes in that test tube, and then you just shake and bake. It is an incubation and oscillation, like you are just agitating and incubating, right? Like that. The way that this, remember the biology of prion diseases, if this comes into contact with the normal form, it will cause it to convert, right? So that's what's happening inside the test tube with RT-QuIC. If this sample is positive, it goes in there, it starts a daisy chain, and these start misfolding, and then it propagates with time. Well, what's really cool is that in the Alzheimer's research area, when looking at amyloid plaques, they developed this thioflavin T, it's called. Just think of this like a little fluorescent dye. That fluorescent dye actually binds quite nicely to the misfolded form. These are like amyloid sheets, just like Alzheimer's plaques. That dye called thioflavin T binds to this. Well, all of a sudden, like if you're shaking and baking, and this is going and it's positive, what happens is you get thioflavin T start binding to that, and then over time, you beam that with a laser, right? So you beam this reaction, find the test tube, you're beaming me with the laser, like every 15 minutes, and if it's positive, you start to detect this curve starts to go up. If it's a screaming hot lymph node, this curve's going to go up in about 9 hours. If it's an environmental sample, it might go up in 20 hours, right? So if you have a lot of misfolded prions going in the sample, it'll go up fast. If it's environmental, not very many, it'll go up slow. So it's essentially a PCR for protein misfolding, and you can observe it in real time. That's what is so cool, is that you can actually go in, if a reaction's been running for a few hours, you can see which samples are going positive, right? So you get information in real time. And that's how this RT-QuIC method works. Highly sensitive, reproducible test. We published this last year, and what this paper shows, and there's other papers out from other groups that are essentially doing what we did here, but with the DNR up in Minnesota, they gave us essentially a brown paper sack that had tissues of 500 white-tailed deer in there. They shook it up and said, hey, we've tested all these with ELISA and IHC. We know there's 12 positives in there. Tell us which ones are positive with RT-QuIC. So we were blinded. We didn't know. So we went through those, and what happened was the RT-QuIC method, we identified all 12 of those, plus 3 more that we thought were indicative of an early stage infection, right? The main thing is here, we did not identify like 200 positives, right, or 300. We found 12 plus 3 more that were likely early stage infection. So we're not seeing evidence of false positive with tissues. This is, on this graph here, this is something called a max point ratio that is a statistic that we like to use when looking at these data. This is over 4,000 RT-quick reactions. Everything, this cloud, this distribution of points here, this is all on the left-hand side of this graph. These are all negative samples that were confirmed negative with ELISA and IHC. And what you can see is the positive samples, you can't even fit them on the same scale, right? It's not like this cloud of negatives is like spilling over here into the positive in this heart. It's like, no, the positives are just like, no overlap. So with tissues, it's pretty exciting because what we're seeing is that it's reproducible. We think that the false positive, false negative rate is as good as ELISA and IHC or better than. So it's very reliable. And the average time to detect like a tonsil or lymph tissue that's positive is 11 hours. This test is so sensitive that you could take 1 tablespoon of these misfolded prions, mix it into 400 Olympic-sized swimming pools. This test could pick it up. So that's great. So RT-Quick is out there. We know that USDA is going through that RT-Quick validation process. They're anticipated to complete that within the year. More and more labs are getting RT-Quick. Well, then we asked the question, okay, so we got RT-Quick up and running. It's working really well. How can we take it to the next level? How can we develop other next-gen tests that would maybe be field deployable? So we had this breakthrough. Keep in mind, everything I'm sharing with you today was done from 2019, so throughout the entire pandemic. We have some really great graduate students in the lab, and one of those graduate students, his name is Peter Christensen. He was working by himself in the lab, October 2020. We know that the normal function of these prions is to bind to metals in different ways. So we asked the question, gold nanoparticles, can prions bind to gold nanoparticles? Why is that important? Every COVID test, the rapid COVID test that you use, a pregnancy test, they're using gold nanoparticle technology. That's what causes the color change, right? When there's a binding event and then the gold nanoparticles are brought together, it changes color, and that's why you see the stripe. So it turns out that technology is used in diagnostics for a long time. So we just asked a simple question, has anyone used like a shake and bake, like a quick reaction followed up with gold nanoparticles? No one tried it before. So the graduate student tried it, and he was by himself in the lab, and he saw, it's hard to see, the colors are different on here, but he saw this color change in the lab, and he's like, holy blankety blank, like this is like incredible. But essentially, and they're washed out on the screen, I'm sorry, but over here, like all the negative samples are like purple, and these are all like bright red, and you can see with your naked eye, just by using gold nanoparticles in these reactions, that they're easily distinguished by eye, and you can measure that also with, but just by measuring light spectrum analysis. Okay, and this is highly reproducible. We've done hundreds and hundreds of reactions with this. And essentially what this MinQuick test is that we developed was you can take the same, everything up to here is the same as RT-Quick, right? So you take the sample, you shake and bake, but then all of a sudden you go in and you just spike with these gold nanoparticles, and the negative sample actually turns blue or purple when there's no fibrils in that test tube. But what happens with the fibrils in that reaction, these gold nanoparticles don't bind to it, and it's red. And all the reactions that we're doing with tissues is that this test is 100% in agreement with RT-Quick results, the IHC and ELISA results on those same tissues. So we're going through a formal validation right now and submitting that to the USDA, hopefully mid to late 22 this year. The thing about MinQuick is, so this test, it's field deployable. It uses cheap equipment. So we were the first team, as far as we know, to actually go out into the field, set up quite literally went into a DNR field station, wiped off 2 tables in the back. There's snowmobiles on this side of the picture. We're back in the warehouse, set up our mobile lab, did RT-Quick, and we got results in 24 hours on site. So you can see how, what potential utilities there might be to make this more make it more accessible to test for CWD. The equipment for this test, it's like $25,000 cheaper than RT Quick. It's only about $3K. RT Quick reaction costs like $15 a sample. This costs like $3 a sample, right? So I don't have time to get into the other technologies that we're doing, but we didn't start, when the state invested in us, we didn't start one line of research on developing next-gen technologies for CWD. We started 4, all in parallel. We didn't have our eggs in one basket because we didn't know which one was going to pan out. Turns out the research that we're doing, those 4 lines, are all providing results. So we think that eventually we can get to a deer-side test, but right now I'm holding this device in my hand. That's not the device that we've developed, the prototype that I'll talk about. This is actually a DNA sequencer, but it's that size of a candy bar. We have a functional prototype that can detect CWD in about 4 hours. It's about the size of that device, size of a cell phone. We're filing a provisional patent on that. So that to say that 3rd generation, like field deployable technologies, like truly field deployable, we think it's feasible and it's on the horizon. We also have this intense research effort to identify small molecules that can bind to the prions in different ways. And it's that research line that we think can actually get us to a deer-side test. We have a milestone around 3 or 4 months out. If we reach that milestone, I really think that there's a direct line there to get to a deer-side test. Okay, so how can we use the technologies that we're developing to help manage CWD out there in nature? Well, another line of work that we started doing was, hey, can we take surfaces, like we know prions hang out, right, for a long time. Can we take certain surfaces and see if the CWD prions are binding those in different ways? And it turns out, yes, there's certain surfaces like steel that prions will readily bind to. And so what we did with a collaborator named Jason Bartz, just a fantastic collaborator at the University of Creighton, is we developed a swabbing technique, and we have these surfaces that we can actually spike them with like about a 1,000,000-fold dilution of prions, swab those, and then test them with our technology, with RT Quick. What this means is we think that we can have sentinels that can be used for real-time herd-level surveillance on a ranch, in the farm, or in the wild. And that you can place these within feeders or troughs near water sources. What we did to test this out is we actually, we were down here in Texas early last November. We deployed some of these sentinels in a positive deer herd right in a feed trough. And the sentinels themselves are about this big, right? So in one trough, you could have like 3 in there. Put feed in the bottom, feed on the top. What happens is the deer come in, and as they're feeding on that, they're licking those sentinels, and so they're getting saliva all over them, right? Left them in that for 1 week. Left them in the pins in that positive herd for 1 week, and then shipped them back to our MinPro lab to swab them. So what's really cool is that this is working. The sentinels, and I know this is data is kind of messy and whatnot, but the sentinels in pin 1 are here, right? And sentinels in pin 2 are here. So basically what this is showing on like 4 different sentinels is these are all screaming hot by RT Quick. Strong positives. In pin 1, 13 of the 19 deer that were in pin 1 were positive by IHC in multiple tissues. So these CWD was in those animals for a while circulating, right? And that's what we saw. These are the screaming hot swabs from the sentinels in that pin. Pin 2 was 1 animal out of 12 was positive by IHC, and we thought that it may be like an early stage of infection based on the data we got back from Texas Parks and Wildlife on that. But it's still statistically positive. So what we're seeing is that 1 out of the 12 animals was positive, that it was still depositing prions, on these 2 sentinels at a detectable level, still statistically significant, but not as much as the herd that was screaming hot. So that means that if you have 1 individual licking on it, we should be able to detect it, that if that individual has CWD, and if you have multiple, we really should be able to detect it. So what does this mean? Well, we think that, and we have a provisional patent on this, we think that this sentinel technology opens the door for real-time surveillance and culling, because you can place these sentinels, right, in a feeder like this, and as they interact with it, you could swab like once a week or something like that and actually test it and then see what's going on in the landscape. Let's say this is your ranch, and these are your feeders, and you start swabbing, every few weeks, let's say. All of a sudden, you get a hit. And you're like, okay, I got a hit there. Is that real? Is it, what's going on there? So maybe you take a closer look and you start swabbing those. All right, here's another hit. Oh, crap, we're getting a lot at that particular feeder. We know now the data coming back is suggesting that there are positive animal or animals going to that. We need to come in here and cull, right? So that's a strategy that we think may actually work for active surveillance and management in real time. We even think that some of these deer come in and feed off of some of these feeders like 6 times a day maybe. You might even be able to pin it down to individual depending on the frequency of your swabbing. So the next generation technologies that I'm talking about, I think that with field deployable diagnostic tests like MinQuick and some of these other things that we're developing, with the sentinels on ranch level deployment in cervid farms. We're also deploying these in the wild up in Minnesota. We're working with our DNR, and we have these out in some of their surveillance efforts. We think that you can actually actively, progressively work to prevent the spread of the disease using technologies like these. Okay, so that's all great. Rapid portable diagnostic technology, sentinels. We've shown that with the funding that we got from the state that we could actually develop the prototypes and get there, so that's all feasible. But what's next? How do you really win the war? Where do you go from there? And we need a multi-pronged approach, and that is surveillance, but we also need to start thinking about therapeutics. Are there ways that we can actually slow down the disease from spreading in various settings? So it turns out that, again, the biological function of normal prions is to help regulate copper, zinc, and these other molecule or metal ions. So nutrition, it's a very, very component of the CWD puzzle. When CWD was first described, it was thought of as a copper disease, that it was that it originated from cervids being in environments with low levels of copper. And that's been in the literature for a long time. But there's papers that are more recently coming out that are showing that prion protein is working, binding nicely to zinc and to copper. And we know that, and there's also literature coming out of the USDA, Tracy Nichols has worked on this, showing that there may be a diet component that is really important for disease management of CWD. So this has been in the literature for a while. So there's actually another body of literature when looking at scrapie that indicate that certain kinds of copper can actually bind to and reduce the spread of the progression, that daisy chain reaction. There are certain types of copper that can actually slow that down. And they're doing this and they've done this in a mouse model infected with scrapie. And by feeding, in their case, I think it was copper sulfate enriched diet, actually significantly slowed disease down. So it turns out that and when you think about copper or zinc, and I think Dr. Hewitt might talk a little bit about this too, but if you think about these additives, it's copper is not copper is not copper. There's different kinds, right? There's different types, and that really matters because you have to have the right type that enter the cells and are uptaken in a different way if you're going to think about ways that can be used for CWD therapeutics. So there's this chelated amino acetate copper and zinc that is produced by Zimpro and that Lycian Echo Feeds uses that it's shown to have significantly upregulated intake into various cells. And what's doing that is, we believe, the prion proteins that are responsible for handling copper and zinc, right? So we hypothesized that these amino acetate copper and zinc could actually inhibit prion misfolding. We're starting off in a test tube model, and I'll highlight some of those data, but we think that we really have to explore this in deer. So we started a collaborative effort with Lycian Echo Feeds who are using these Zinpro supplements. And again, these data can be kind of messy, but we asked the question quite simply, and this is replicating what some others have done, again, in Scrapie, and that is like, can copper slow down or inhibit the misfolding of prions in the test tube model? And what we found was that, yes, actually there's certain combinations or dilutions of this amino acid copper that spiked in the RT-core reactions. You can tell like a 2-to-1 ratio. It's like 2 parts copper, 1 part of the normal prion protein. It actually slows it down versus positive controls. And as you get really, really more dilute, then you have, there's no difference between your positive controls. What that's doing is actually, this is screaming hot positive controls. The rate of amyloid formation, which is like the way that those molecules misfold, those slinkies start to misfold, that is slowed down, and it takes a lot longer for it to happen. Okay? So that's what's happening with the copper in vitro. The zinc is really interesting because it seems like there's this titration effect, and at the highest levels down here, it was also shifting these graphs over, reducing the speed, if you will, of the misfolding in vitro. So this agrees with the work that was done in Scrapie, and we think there might be something worth looking into with live animal studies. So that was like a milestone 1, and now we want to work with Zinpro and listing nickel feeds and try to get into a cervidized rodent model. So we can do this at Creighton with our collaborator Jason Bartz. We can actually have an enriched copper and zinc diet, and then it would be fantastic if we could find a way to actually look in a very sophisticated scientific manner if in a deer herd, if we can slow down, right, that rate of CDB progression in a positive herd. So our hypothesis is that you can inhibit prion misfolding within an individual, and that might actually be able to effectively reduce CWD transmission through direct and indirect contact. Why is that? Because if the disease is spreading through an animal, and if it's unrestricted, it could be outputting the prions in the environment in its feces and urine. Like, it could be like a super shedder, if you will. If you slow that down enough, maybe it's not outputting that in the environment, right, to a significant level as without inhibiting the prion misfolding. That's really important, so we have to understand that. Another reason why it's important is that if you were to slow it down, you may actually come in with another molecule and knock it out, right? So you could actually design small molecules that are like heat-seeking missiles that could, if you time this all right, you can come in and actually knock out the misfolded prions. Okay, so we think that actually winning the war against CVD, it's going to require a proactive, next-generation, multi-pronged approach, and that's what our center does. It really brings together folks in different backgrounds trying to work and solve this problem. We think that faster portable diagnostics is really essential because you could have early detection sentinels, like a ranch level, and then you can go in and targetedly call and remove those animals. We need science-based nutrition for a healthy deer population. That, again, in the literature, this is an important part of the CWD puzzle, and it's been understudied, and we need to do better at that. And we're working right now with one of our lines of research that can, on these small molecules that can actually bind to and neutralize this misfolded slinky here. We can actually bind to it, we hope, with these new molecules and prevent the misfolding. So that's a different line of research that, again, 3 to 4 months milestone we should know about. So many folks to thank. Thank you so much for inviting me, and I know the whole Minnesota team is happy to be down here, and I'm happy to be back down in Texas, and lots of folks to thank. Lissy and Echo Feeds with Ronnie Eckel, Mitch Lockwood, Texas Parks and Wildlife, and of course, Dr. Hewitt and the rest of the folks at the center. Thank you so much. Open for any questions.